what is our topic? The story of microorganisms. Okay, so story in the sense, what are the things that are involved in the microorganisms that we need to know? We will discuss in this session about the what are microorganisms, okay, and how many kinds of microorganisms are there, and why these microorganisms, whether they are harmful or beneficial, or how many kinds are present in how many organisms these microorganisms are present, right? So we will discuss and we will answer all these questions. Okay, so first of all, the basic thing that we need to know about the microorganisms is the first basic question that we will get in our mind is, what are microorganisms, right? So I'll give you the definition for this. So, right? Yes, I'll make it two parts of this word, right? Micro. What is the meaning of micro? Micro is very small, very fine, smallest thing, right? Micro is used as a measurement for the small things, right? So, what is organism? A living thing, right? So, what is the definition for microorganisms? What microorganism mean, right? Microorganisms are the smallest living cells which are not seen with the naked eye. I'll put it on the board. Microorganisms are the smallest living things which are not seen or observed, which are not seen or observed with the naked eye. Right? I am underlining the point naked eye and I am underlining the point smallest, and you all know that seen also. Why I am underlining all these things? Because smallest living things which are not observed with the naked eye. Naked eye in the sense real eyes, right? With common like this, I can observe the chalk with my naked eye because this unit is not that much fine, small unit. So it is visible to us, right? Very clearly, the thin part. If I make it as a powder, so I'll drop this powder. You are observing that? Are you able to observe this? No, right? So how difficult it is? Likewise, if a hair follicle, I'm just taking one hair follicle. Are you getting this hair follicle? Are you observing this? When I take thin section of this hair follicle, it is very difficult for us to observe, right? So, microorganisms are also a kind of this category. So, how we will observe these microorganisms? Microorganisms are observed with the microscope. Okay, we'll go to the discussion later. Then, who are the scientists? They laid the foundation for the discovery of microorganisms. The Anthony von Leeuwenhoek. In the cell, we have discussed that Anthony von Leeuwenhoek in the year 1657, he laid foundations for the discovery of the cell. So, likewise, he invented the simple microscope with 100 and 300 magnifying lens, so that he observed the smallest living cells, smallest units. Those are called as the cells. So here also Anthony von Leeuwenhoek is considered as father of microbiology. Okay, the study of microorganism study is so called as microbiology. Okay, the study of microorganisms is called as microbiology. So he is considered as father of microbiology because he discovered the microorganisms right first, and in the past lesson we have discussed that he named them as animalcules because as they were moving like an animal, he named these microorganisms as animalcules. So we can say that he is the scientist behind this discovery and the story of microorganisms. So it is so aptful, right? So who is the father, who is the discoverer for these microorganisms? Anthony von Leeuwenhoek. Anthony von Leeuwenhoek, right? Father. He is the father for the story of microorganisms. So what is the story? The story should have an introduction, the person behind it, right? Any kind of story, if you are reading, right, it should have some introductory part, I mean the past things that happen, and also who are the persons behind that story, and what is the main theme of the story, and finally the conclusion. So in this microorganism story also we will discuss we have discussed the definition, we have discussed the father or hero for this microorganisms. So now we will discuss about the various kinds of microorganisms that are present in our surroundings or in the surrounding media. Can you classify the microorganisms? Are you able to classify it? Yes, I'll let you know how to classify the microorganisms. Basically, the microorganisms are classified into five kinds of microorganisms are present in our surroundings. Okay, so what are that microorganisms? Basically, I'm going to classify here. Microorganisms are broadly classified into five kinds. You know, bacteria, number 1, and 2 is algae. I'll tell you what are these all. Fungi, right? And protozoans, and finally the viruses. There are five different kinds of microorganisms. They are bacteria, algae, fungi, protozoans, and finally the viruses. Microorganisms are divided into these categories according to the type of organism, right? They are classified into 5 different groups. I'll give you one example to understand this. Take for example we or any kind of animal, right? We are different from the animals. And the animals are broadly classified into domestic animals, wild animals, right? And we will differentiate the organisms like omnivores, carnivores, herbivores. Herbivores are the grass-eating one, so they are categorized into one category because of their habitat and food habits, right? So likewise, carnivores are one kind of thing, like they will eat the flesh. So they are grouped in one category. They all belong to one group. And whereas the omnivores, they are classified because they will eat both the grass, I mean the plant source they will depend upon, and apart from that they will also dependent on the animal source. So that's why they are the omnivores, right? So we are all the omnivores. And animals, some are domestic and some are herbivores and some are the carnivores, and sometimes some are omnivores too. So like this, how we have differentiated the organisms based on their food, their habitat, and their actions, right? So likewise, here are the smallest things, the smallest microscopic units also, these microorganisms, they are broadly divided into bacteria, algae, fungi, protozoans, and viruses. I'll give you the live and the most common examples for this. Take for example this bacteria, right? What are bacteria? Bacteria are one kind of the microorganism, right? Take for example your skin. While you're touching untidy or dust particles, right? So you have, I have fallen down, right? So here the soil contains different kinds of bacteria. So immediately when I observe my hand or when I take small scraping of my skin, and when I observe it under the hand lens, I mean the microscope, I will observe different kinds of germs are rotating or moving on my skin, right? So bacteria is so familiar to us. And also the algae. Algae is most commonly observed in nearby, the most common observed algae is near the ponds or the tanks, right? In the tanks or in the ponds where the more humid conditions are present, wet conditions, there you will observe soft green color material, right? You will call it as in Telugu you will call it as nachu, right? So that green algal growth, it is a green plant growth. So green planktons we will call it as. So that green algal growth is so termed as a algae, right? That you will observe in your nearby ponds or you in the lakes, and sometimes most frequent you will observe in the aquariums too, right? It's because it is the most suitable conditions for the growth of the algae. It is a living thing, right? So it lacks all the other like cell organelles in the algae. So it is one kind of living thing with the plankton, right? So that growth is so rapid here. And coming to the fungi, have you ever observed the spoiled food or any other kind which is spoiled, which is expired or which is spoiled? Take for example one potato. After 7 days when you observe the potato, it gives a very foul smell, right? Why the foul smell or rotten smell? Spoiled smell is coming out due to the growth of the fungi there. The fungi grows there and thereby you will get the smell, that what you call it as very foul smell, very rotten smell, right? So that is, and powdery things you will observe in the benches, right? The powder in the corners of the room, right? And small rug-like structures where the it looks like a rug-like structure around the bench or in the corners of the room or in the windows, or when you keep the books for a long time, when you're keeping the books on a side for a long time, there you will find small microorganisms coming and they're forming a rug-like structure like this, right? With all sand particles and powdery forms. And sometimes when the bread is spoiled, you will observe the powdery material that is coming out of the bread in the form of green or in the black pepper colored spot like structures. That is also one good example for the fungi. And coming to the protozoans, you are so familiar with these protozoans, right? So that protozoan is what? Amoeba. In your earlier classes, you are coming across this word. This organism comes in every chapter, right? So it is an irregular organism. So I'll write here with the examples too, so that it will be convenient for you. Amoeba, right? I'll draw a small diagram here. It is an irregular organism, right? And also the paramecium. So here mouse, bread mold I'll write here when it is get spoiled. So it is one good example for the fungal growth. And for example green pond algae, here I'll write green algae in the pond, right? And for the bacteria I will write the germs as one good example, right? And coming to the last one, this is virus. Now it is so rapid, the virus, right, that is surrounding us, that virus is coronavirus. So I'll write here, that is one good actual example for, right? So since from your childhood or many years onwards, we were discussing about various kinds of viruses, right? So the measles virus, the Ebola virus, the swine flu virus, the now fresh virus that is surrounding us and that is troubling us a lot. That virus is nothing about the coronavirus. You can take it is also constituted to a kind of the microorganism. Micro or microorganism, both are similar. Okay, one kind of microorganism that is troubling a lot in nowadays is nothing about the coronavirus, right? So these are the classification. This is the classification for the microorganism. So we'll do some activities and we'll perform some activities to understand this one by one. How the bacteria will be, how this bacteria is developed and how this algae is developed. When you observing that the kind of cells, the kind of microorganisms in the algae, how they are, and also the fungi, how the structure of the fungi, how is the structure of the fungi, how can you collect, how can you strain it on the slide, that we will also discuss. And the protozoans, amoeba and paramecium I can show you, right? And the virus, I'll give you a number of structures of the virus, okay? And what these microorganisms, what they'll cause, what they lead to, what is the way, what they will depend upon. So that we're going to discuss now a bit detail, okay? So First of all, we'll discuss some common examples that will daily we will come across this, right? I will ask you some questions also, right? Right? So, have you ever observed how the food is getting spoiled? How the cooked food gets spoiled rather than the raw food? Take for example the vegetables. When you are exposing to the outside environment also, they will not be getting spoiled. How this happens? But whereas the food that is preserved or it is cooked and it is stored in a container, right? So it gets spoiled at the end of the day. When you are cooking in the morning, when you take the food out of the box or out of the container, at the end of the day, like evening, the food gives you a false rotten smell. How this happens? Why this is happening? Because here the microorganism starts replicating, means that they are dividing and they will increase in number, so that the food at some point it will not resist this growth of microorganisms, and thereby the food gets spoiled due to unconditional growth of these microorganisms. Take for example the carrot, the potato, the onions. For till 6 days, when they are not cooked, they are fresh and they are edible, and then you can consume them. Whereas the cooked food which you are cooking in the morning and when you are consuming it in the evening, when you are going to consume it at the evening, you are not going to take the food inside. It is not edible. It is non-edible. Why it is non-edible? Because the growth of microorganisms here increases. That condition, that humid, I will write here, growth of microorganisms means the bacteria. Which bacteria? Growth of microorganisms means the bacteria, right? So the bacteria starts increasing. Bacterial growth results in, okay, so the bacterial growth results in rotten or foul smell. Means here, what happened here? Here, the food gets spoiled. Spoiled food. Right? The food is spoiled here, and why it is spoiled? Because of growth of microorganisms. Means here one kind of microorganism, that is the bacteria, it start increased. The growth of microorganisms here increase. The bacteria increases in concentration, increases in number, thereby it results in releasing the foul or the rotten smell. Right? So this is the kind of the bacterial growth that you will observe in the spoiled food. Right? Why then? What is the difference between the cooked and also the uncooked food? Why the cooked food gets spoiled soon, whereas the uncooked food, here is the answer for that. Because here that condition, that humid, that wet conditions and closed, wherever you will find the closed humid conditions, there that is the main site for the growth of microorganisms. What is the main site for the growth of microorganisms? Wet humid conditions. Right? So, wet humid conditions is the most suitable condition for the growth of microorganisms. Here, the closed environment which is separated from the outside environment will make the bacteria to multiply, and so that it will increase in number, so that the growth of microorganism here happens. What is the kind of microorganism here in the cooked food? The bacteria. So this bacteria will multiply in number, thereby the food contains a moisture, right? Whatever the food, that is a fruit, the vegetable, the other kind of foods, when you just cut them, when you observe it, potato, when you cut it, you will get the water. The cucumber, you get the water, right? So what all the minerals, the water, the salts, they will be coming outside it, right? When it is cooked, the water, the materials that will come out of it, and thereby the wet conditions, the closed environment will increase, and thereby they will multiply here, the microorganisms, and ultimately it results in the bacterial growth, and also that's why when they are increasing, you will get the rotten or foul smell. You know, bacteria will multiply in the numbers in the manner of a square. Take for example 2 square, 4 square, 16. Like that, in the fraction of seconds, in 1 minute, they will be of increasing and bulging in the nature, so that will give you 4, 4, 2, 8, 8, 8, 2, 64, 16, and 16, 32, 8, 64, and thereby they will multiply within a second, so that the growth is so rapid, so fast, you cannot stop the growth. So that's why here the cooked food gets spoiled soon than the uncooked food. Okay, so now we'll discuss about this. Okay, take for example, and I'll also give one more example also. The curd. Have you ever observed and you have tasted the curd, right? Daily, it is a daily routine for us. So how the curd is formed? How are you preparing the curd like the rice? No. And then how is the curd is formed? The curd is formed when you are adding 1 drop of buttermilk or the curd to the freshly boiled milk, right? So, and we will discuss about one thing also. Why we will boil the milk? The milk is boiled to kill the microorganisms, and this procedure is so called as pasteurization. Because of the scientist, it got the name Louis Pasteur, pasteurization, right? So here we have killed the microorganisms, but not completely. Here we are also having useful microorganism, that is bacteria in the curd. What is that bacteria? Lactobacillus bacteria. Okay, I'll write here. So the lactobacillus bacteria, it will result in formation of the curd. Okay, so how the curd is formed? Curd is a composition of lactobacillus bacteria. Okay, lactobacillus bacteria. So this lactobacillus bacteria, it will increase in number, and so they will multiply, they undergo the division, and thereby the liquid part, initially the milk is in the liquid condition, right? So the milk contains the microorganisms. First of all, why after extracting the milk from the cow, we will just boil it. Why we are boiling here? To kill the microorganisms at 30 degrees centigrade, right? So at that heat conditions, what it happens here, the microorganisms, some sort of harmful microorganisms are killed here, and but the milk is not losing its useful bacteria. Okay, so initially the milk will be in the liquid form, right? It is a liquid in condition, and after a while, when you are adding 1 drop of buttermilk to it, it turns into homogeneous solid, right? Homogeneous form means the solid form. Homogeneous means uniform concentration, right? It is uniform, thorough concentration. It is uniform in the manner, in the same sense, right? So how the liquid gets turned into the solid, how it is getting turned into the solid due to presence of the bacteria, that is the lactobacillus bacteria. It makes the liquid milk into the curd. When you adding the buttermilk, what is the buttermilk? Buttermilk is also a composition of the curd. When you just making it when you mix it thoroughly, when you stir it thoroughly, that homogeneous liquid, homogeneous solid form is turned into the liquid, right? So when you just adding that 1 spoon of curd or 1 spoon of buttermilk to this homogeneous liquid form, the milk, it gets turned into the curd. How this is the preparation of the curd, right? So everyone know this, but the story behind this turning of this liquid to solid is because of what? Because of microorganism. What is a microorganism here? Lactobacillus bacteria. So eat this good bacteria which is good to the health. Okay. So when you adding this to the milk, it is just turning. It is means undergoing the division. It will multiply, and the bacterial growth is completely over. At the time, it turns into the homogeneous form, that is a solid curd. Okay. So this is the preparation behind the curd, and sometimes after 1 or 2 days, the curd gets spoiled, isn't it or not? Right? So it gets spoiled, and you will get very rotten, as like in the vegetable or fruit manner. Here also you will get the rotten and also the foul smell. So here we will collect here that material and we will test it under the microscope. For what? To observe the bacteria. So I will take the spoiled curd. Okay. in spoiled curd or the buttermilk you may take here for the experiment, observing the bacteria in the curd, because bacteria contains the lactobacillus bacteria. Okay, so that's why I'm going to take the spoiled. You can take here the curd or you may take other spoiled food also, but I'm taking here the spoiled curd here. So first of all, what I'm going to do, I'll take small drop of the water from the curd. So as like we have performed in the previous experiment, like taking the glass slide means one glass piece, right? So as like in the same manner, I will take this drop of buttermilk or the curd on the glass slide. So immediately for a while I'm going to heat it, or I will observe without heating also. I will add some indicator. Why we will add this indicator or colors, or how we are staining this, why we are staining this? To observe the contents clearly. Some areas of the cell or any kind of organism which is not visible with our naked eye or in the microscope. To observe it so clearly, I will add here the stain. So here I will add the stain as crystal violet. Okay, crystal violet or methylene blue. Sometimes you will use the red ink also. So what I'm going to do here, to this foil curd, I will add the indicator also, and slowly after adding the indicator, I'm going to wash it and I'm going to heat it. Okay, so that the impression will be present on the glass slide. Okay, so and finally I will cover it with the cover slip that you have to perform in every activity, right? So I will cover it with the cover slip. What I'm going to do later, I'm going to observe it under the microscope. Later I'm going to observe it under the microscope. I'm going to observe the bacterial growth like this. Okay, the bacteria are of different shapes, like how the cells are of different shapes and size. The structure and functions are different. Likewise here also the bacterial shapes are not confined to only the spherical, the columnar, the oval or any rectangular shape. The bacteria of different kinds, the shapes and sizes, they are varying here. Here also when I'm observing this slide under the microscope, I'm going to observe like this. The cells may be of like this, round or rectangular or cylindrical or pointed, right? But I get some cells. So these kind of organisms are called as bacteria. Why we have observed this bacteria? Due to the curd, when it is spoiled also, there you will observe the growth of microorganisms. Why? Due to presence of the bacteria, which is so called as lactobacillus bacteria. So we have finished the one part, one kind of the microorganism, that is bacteria. So means that the bacteria is also a kind of the microorganism and it is present in all the surrounding mediums. One good familiar example we can use here is the spoiled food or also the curd or buttermilk you may consider for the experiment. So we will also perform another experiments later to observe the fungi, protozoa, viruses, right? And what either they are harmful or beneficial, either they will result in diseases or not, and what precautions we need to perform, that we will discuss later. I hope you understand this lecture.